Thank you for that. Now we have our third presenter, Humaira from India, and the title of presentations will be uh, proteomic characterizations and antibacterial synergy of coliphage-derived proteins against MT or E. coli. Over to Humaira. We can see your presentation.
Hello, Suraj.
Good evening, everyone. I am Humaira Saeed from India, a PhD scholar working at
Humaira, can I stop you? Sorry.
Okay.
Yes, Asifa, please. Could you please interrupt after finishing her presentations? Thank you, Asifa. Over to you, Humaira, again. Sorry about the interruptions.
Once again, good evening, everyone. I am Humaira Saeed, PhD scholar working at Amity University, and I'm from India. So today my topic for presentation is proteomic characterization and antibacterial synergy of ASEC2201 coliphage derived proteins against a multiple drug-resistant E. coli. So beginning with the rationale of the study, as we all know that antimicrobial resistance is a growing global health crisis as quoted by WHO as well. So for this, we need sustainable solutions to tackle this very problem. So for that, the solution has to be very specific to bacteria to overcome this problem of resistance. It should be effective against the MDR strains, as well as it should reduce the antibiotic dependence, and it should be eco-friendly and safe. So in that light, we have bacteriophage therapy. As we all know, bacteriophages are viruses that parasitize bacteria, and they are present almost everywhere, such as from glaciers to ocean bed, etc. So the use of this phage therapy is not very new. It was recognized in 1921 by the biologist Felix de Herel. He reported success using various phages to treat a variety of ailments, including cholera and plague. So phage therapy has generated considerable interest since then due to growing incidence of AMRs. So now this phage therapy can be used in many ways as shown in the picture, such as it can be used as monophages, single type of phages can be used for the therapy. Then different phages in a cocktail form can be used as well. Then phage antibiotic combinations, which I'll be talking about in this very presentation. And then we have a specific phage
Sorry to
enzymes.
interrupt again, Humaira. Sorry. Can I ask Asifa to stop presenting because we can't see Humaira's presentation. Instead, I can see yours. Thank you very much. Thank you, Humaira. Please continue.
Okay. So then we have phage enzymes, which can be used for this phage therapy, such as endolysins, then spannins, etc. And then we have engineered recombinant phages for this very therapy. So in this presentation, we'll be discussing about phage antibiotic combinations. So in this study, we have basically characterized a phage against Escherichia coli, which is ASEC2201. And then we have done the proteomic characterization through wet lab techniques such as SDS phage and then 2D gel electrophoresis. Post which antibacterial activity testing of that very phage is done, in which we have determined the MIC breakpoints and we have seen the time kill kinetics. And then we have performed the synergy with antibiotics and we have seen the bactericidal effects of the very same phage. Proceeding further. So these are the results. In first picture, you can see the clear lytic plaques of the ASEC2201 phage. And then this phage was ultracentrifuged and we have done the TEM analysis, which revealed that the phage belonged to the Cyphoviridae family. It is a tailed phage. And then the phage was precipitated through PEG and acetone to have the protein precipitated out of which. So the phage protein was precipitated and then the molecular characterization proteomic profile was done using the SDS technique and the 2D gel electrophoresis. So in this first picture, as we can see, the first lane is the E. coli proteins, just to demarcate between the phage protein and the E. coli protein profile. So, and the second lane shows the ASEC2201 proteins. These are the precipitated proteins. And then we have in second picture, we have the molecular weight marker in the first lane. Second and third lane is ASEC2201 whole proteins. We have got a band near 18 to 20 kDa, which was re-eluted and it was rerun on the gel, and then again this purified band of the lytic protein we have got at this position. This very molecular weight was verified with the in silico characterization, as we have done the genome sequencing of this very data also. So in that physicochemical characterization, the lytic protein molecular weight was around 18 to 20 kDa, as we have got in this gel. And then we have re-eluted it by dissecting it from the gel and then again run on the SDS. So we have got the sharp band at 18 to 20 kDa. And then further, the 2D gel electrophoresis revealed that the same spot at 18 to 20 kDa was at 9.5 pH range, because phage proteins are basically basic in nature. So this was also very further validated protein was a lytic protein. Moving further, so we performed the time kill kinetics of the phage protein in first graph. This is solely the host, which we have seen over time the growth, and as you can see, it has grown to 8 log growth. Full growth is seen after 14 to 16 hours. In this, the second graph, we have treated the same host with the phage protein at the MIC point, and the MIC point was further diluted. So just to check the kinetics across the time. So as you can see, as the time is increasing, the log is decreasing with time. As you can see, the bars, so the growth was inhibited. This showed that, and then we performed this diffusion assay as well, in which we use 3 beta-lactam antibiotics of different generations: cefepime, imipenem, and cepedroxil. So antibiotics itself served as a control, and it was also treated with protein with antibiotic, the phage proteins. So antibiotics alone showed 28, 38, and 31 mm of ZOIs for the 3 antibiotics, but when it was treated with the protein, the ZOIs increased, such as for cefepime, earlier it was 28, but with the protein treatment, it was 35 mm measured. And for imipenem, from 38 to 45 mm, the increase was seen, and for cepedroxil, 31 to 42 mm. So basically, MIC and this ZOI assays confirmed that the protein showed a lytic activity, and hence it can be concluded that morphologically the TEM analysis revealed that the phage belonged to the Cyphoviridae family, and SDS phage analysis revealed distinct phage protein bands within 20 to 66 kDa, indicating multiple structural and lytic proteins. 2D phage profiling showed a broad PI distributions with 3 to 10 PIs, with several proteins localized in the basic region, as already told. The phage protein fraction demonstrated significant antibacterial activity against the MDR E. coli with an MIC of 0.841 mg per ml of the protein, and time kill kinetics showed concentration dependent bactericidal activity with 3 log reduction in bacterial counts within 12 hours of incubation. Combination assays with antibiotics of different generations, such as cepedroxil, cefepime, and imipenem celastin showed the enhanced antibacterial efficacy, and the ZOIs increased by 80 to 35%, which are indicative of synergistic activity of phage proteins with antibiotics. So with this, I come to the end of the presentation. Thank you so much.